Hello everyone and welcome to Lesson 4, Part 3, the last part of Lesson 4. And today we are going to learn how to build a computational model that finds the optimal harvest rate for a fish species dependent on biological parameters of the specific to the species and understand the relevance as to why this modeling is important. So let's get into it. Okay. So fisheries play a crucial role in providing food and livelihoods for millions of people worldwide. However, improper management of these valuable aquatic resources can lead to overexploitation and ecological imbalances, threatening the very populations they seek to sustain. To address this challenge, researchers and policymakers are increasingly turning to computational models to determine optimal harvest rates. At the heart of these models are biological parameters specific to each fish species, such as growth rates, reproductive patterns, mortality rates, and carrying capacities. By integrating this data into sophisticated algorithms, scientists can simulate the dynamics of a fish population under various harvesting scenarios. The goal is to find the sweet spot, the optimal harvest rate that strikes a delicate balance between the sustainable yield and the population stability. When the harvest rate matches the natural replenishment rate of the fish, the population remains homeostatic, allowing for continuous productivity without depletion. Computational models can assess the impact of different fishing intensities over time, offering invaluable insights into the long-term consequences of specific harvesting strategies. By accounting for biological uncertainties and environmental fluctuations, these models enable policymakers to make informed decisions that promote responsible and sustainable fishing practices. Ultimately, harnessing the power of computational models empowers fisheries management to avoid catastrophic collapses of fish populations while maximizing the yield potential. These efforts not only safeguard the health of marine ecosystems, but also secure the livelihoods of fishing communities and contribute to the global food security in an ever-changing world. So overall, this script allows for the investigation of how different harvest rates impact the fish's population, expected yield, and variability. It demonstrates the use of computational modeling to explore ecological dynamics and informs decision-making for sustainable fishery management. So next is the explanation of the script. So this RStudio script is a simulation and analysis of a simple ecological model that represents the dynamics of a fish population subject to harvesting. The main purpose of the script is to explore how different harvesting rates, H, affect the expected yield and variability in a fish population sizes over time. So before we get started, let's look at the homework. So the homework for this is you're going to change the per capita growth rate. So per capita just means relating to each individual. So what is the average growth rate of this species? And we're going to use the per capita growth rate of the bluefin tuna, which is the most commonly fished tuna as of 2022, and that per capita growth rate is 0.286. You also change the spread and birth rate to 0.2. Tuna is very well studied, and due to this, their spread and birth rate is smaller as there are mass data sets calculating the birth rate to a relatively precise spread. And you're going to change the starting population to 100,000. You're also going to change the harvest rate vector from 0.05 to 1 by increments of 0.01. So you're going to test more harvest rates than we will in this example. So when you do your homework and you run this simulation after you've changed all the variables, it will run for a while because you're going to run 0.01 from 0.05 to 1. That's like 100 harvest rates that you're sampling, and for each harvest rate you're doing 500 simulations of 100 years, and so it's going to take a minute to process. So when you click OK and run on this, don't be surprised if it takes a while. So back to what we're going to do. So the first thing we're going to do is we're going to set up our models and our model parameters. So first we have R, which is the per capita growth rate, and for this example fish is 0.5. We're also going to define R spread. So this is the variability in this R value. So this is a pretty big spread. But this is just what it is for this imaginary fish we're doing. And then for this imaginary fish, we have a mortality rate of, what is this, 0.005%. And then our target harvest rate is, we're just going to define for now as 0.05. And that's actually going to be, 5% is going to be our lowest harvest rate. And then our initial, so our initial population size is going to be 10,000, and we're going to run each model for 500 iterations. And so the more iterations that we do with this random sampling, the more certain we can be about this random sampling. So let's just go ahead and get started. Okay, so the first thing we need to do this is we need to set up the treatments and data sheet. So we're going to call this just data sheet. And we're going to use a data matrix. We're creating a matrix, and we're going to define how many rows it's going to have. It's going to have 0 rows for now, and it will generate the rows as we run our for loops. But it's going to have 3 columns, and we're going to define those column names. So we're going to use the function column names, and what data are these names for? Well, it's for the datasheet matrix. And we have to concatenate those because this is a sequence, so we know what order they're in. So the first one is going to be the run number, and then the harvest rate, and then the yield. The next thing we want to define is the treatment, which the treatment is the vector of harvest rates. And so basically what we're doing here is we're going to create a sequence that says you're going to sample harvest rates from 0.05, so 5%, to 100% by increments of 0.1. Okay? And that's it for this section. So for the next section, we're going to just set up our for loop. So again, remember the syntax. For I in 1 through length, right? But we're going to call I choose H in 1, because you can call I anything. So for choose H in 1 through length of HVEC, because HVEC defined how many different harvest rates we're going to have. And then curly brackets, open that up. You are going to define that our H variable is actually going to be HVEC. So that H variable we defined above right here, now we're saying instead of just 5%, I want you to do the entire sequence of 0.05 to 1 by increments of 0.1. And then I want it to choose H. So I want each iteration to choose one of these vectors, right? And then we are going to actually do a nested for loop. So we are going to say for I in 1 through runs, open curly bracket, So I just want to reiterate this. So we are choosing a harvest rate from 0.05 to 1 by increments of 1. And what I'm going to do real quick is I'm actually going to print this HVAC for you so you can get a better idea of what's going on here. And we'll just look at that real quick. So HVAC is 0.05, 0.15, 0.25. So you see how it's starting 0.05 to 0.95 by increments of 0.1. So it's going to test each one of these harvest rates. And for each one of these harvest rates, it's going to run a simulation that spans for 100 years. And then it's going to do that each harvest rate for 100 years, and it's going to run that 500 times. And so this is to create a robust simulation so we can get as much accurate data as possible. So you'll see how this plays out because this is several nested for loops in one another to do this. So we say for I in runs, so I in the 500 runs, there's going to be a time series matrix. And that matrix is going to have, it's going to start with 0 rows and it will add to it for each run. So it will end up being 500 rows, but it's going to have 3 columns. And we need to define those column names. So we're going to say call names for time series. Those are going to equal, remember we have to concatenate it so it knows which one goes to which column. So the first column is year, then the second column is N, so what the total population is, and then the yield. So we defined N initial up here, right? So we're going to tell it that from now on, I don't want to call it N initial. I want to call it N because of simplicity. So I'm going to say N equals N initial, right? And this sets the population at time 1. And then, so we do another for loop now, and now we're saying that for t in 1 to 100, so this is a fixed number of years, so for 100 years, right, we're going to say that H actual, so the actual harvest rate is actually different from the HVEC. So the actual harvest rate is you take the HVEC value that it chooses and then you say it's going to be minimum H times N, where the total is N, where the total possible is N. And then we're going to put the output from this for loop back into the time series data frame by using the rbind function. And so we're going to tell it to rbind the time series data frame. And what we want it to rbind is we want it to rbind each row. There needs to be the year, the population, and the H actual. So the actual harvest rate. Because fishers try to harvest at whatever rate, but there's variability. And so we also need to account for this variability in, oops, I'm missing, there we go. So we also need to account for the R actual. So remember that R is the per capita growth rate. So that's just the average. So we need to get what the actual value probably is. And so to do that, we're going to tell it that the maximum R value, R actual value, is based off a uniform distribution where the maximum ranges from 0 to this uniform distribution, which goes to 1, and the minimum is R minus R spread, and the maximum is R plus R spread. So this is basically saying that the actual per capita growth rate, it will be a uniform distribution from anywhere of 0.5 minus 0.5 to 0.5 plus 0.5. So it's saying that the per capita growth rate is going to range from anywhere from 0 to 1, essentially. The next thing we need to do is we need to define n in this. And so n is actually going to be the initial population plus n times r actual, so the per capita growth rate or birth rate, right, minus mortality rate times initial population times n minus h actual. Oops. So this is saying that we're subtracting the initial population times the amount of the actual harvest rate, also times the mortality rate. So remember back to our SIR model, how you have to, when you update your population turnover, this is what this is doing. This is updating our population turnover. It's saying that it's going to be the growth rate, the spread in the growth rate times the initial population. And so this is going to be a small percentage, and we're adding it to the initial population. Versus here, we are timesing the mortality rate and the harvest rate, which are being removed from the population, and they're being subtracted, right? With the subtraction sign, it's subtracting it. And this is what turns over the population and advances it 1 year. And so it will do it for years 1 through 100. Then we're going to close oh, I forgot to add a curly bracket here. And so I'm going to close this, and then I'm going to add the close down there. Okay, so I needed 3 because we have 3 for loops. So 1, 2, 3, 1, 2, 3. Perfect. So now we're going to go, we finished this inner nested for loop because this is how the population turns over every year. So you can think about it like this. This inner for loop is how the population turns over every year. This says what is my yield in this population turnover considering the H actual. How does this compare for 500 runs? And then this for loop is saying for those 500 runs, like you need to do 100 years of 500 runs for each HVEC value. Okay? hopefully that makes a little bit more sense. But the next thing you want to do is you want to get outside of the inner nested for loop, because now we're going to say, how are we going to spit out this data? So how we're going to spit out this data is we're going to refer back to our data sheet, which is this one that we made, which was for each run, age, and yield, because we're outside of the inner for loop, and now we're in the runs for loop. So we're saying for all of the 500 runs, right? I want you to populate this data sheet. I first need to do the rbind function. But I want you to populate this data sheet, and I want you to populate it with the run, the harvest rate, and the mean of the time series. So we're saying the mean from years 1 to 100, what was that harvest rate for each run? So remember, it does years 1 through 100, that's one run. And then you're going to do that 500 times. So I is one of the 500 times. H is the harvest rate that is chosen from the HVEC. And then mean time series. And then we're going to say from 50 to 100. So we're looking at what was the mean population size, or yield from years 50 to 100. Okay? And we're looking at years 50 to 100 and not 1 to 100 because we're going to see changes in the overall population after a significant amount of time, so like say 50 years. So we just want to see what happens after a significant amount of time. And that's why we're only looking at the mean from 50 to 100, because looking at those earlier years can mess up the mean of the overall dynamics of the population. And this is essentially what we're going to be plotting. So the next step is to go to the plotting thing. But it's not exactly what we're plotting. So we are going to create this thing called datasheet to plot. And this is just simply our data frame datasheet. Okay? We're changing it from a matrix to a data frame. And that's so that way we can do statistics with it, because a matrix is just used to store data. You need a data frame in order to manipulate data. So next we're going to change the, we're going to analyze what the expected yield is by aggregating, using the aggregate function. We're going to look at the yield considering the harvest rate. The data we're going to be looking at is the data sheet to plot, and we want the mean. So the expected yield is going to be our mean. Now, the spread in yield, or the variability, is going to be the standard deviation. So we're going to aggregate again. So the yield considering harvest rate. So what is the yield grouping per harvest rate from the data sheet to plot, and then ST, standard deviation. And then we just want to name this new column. So we're going to say call names, spread, and yield. This is the second one. This is the second column, and we're going to call this, yield bar. So yield variation. Okay, so the next thing we do is we plot. So plot our expected yield, which is the first column in our data frame. So we're going to say all rows, first column. Our expected yield is our x value, versus our expected yield second column, which is actually the spread in yield. That's going to be our y value. And then we need to define the different variables for the actual graphing window. So like our x label, that's going to be the harvest rate. The y label, that is going to be our expected yield and variability. And then LAS, this is just like how big do I want my window. I just want it normal. PCH 16, so this is how big do I want my data points. I want my color to be oh, I'm so sorry guys, I messed up. So when I'm saying my x and y values, so my x value is actually so this was datasheet. So my x value is h. My y value is yield. Yes? I think so. Or I could have it the other way around, because expected yield is where we aggregated yield by harvest rate. So we said y lab is expected yield and variability. Yeah, so the y value is going to be the yield, the x value is going to be the harvest rate based off of how the aggregate function works. So sorry about that, guys, but it will make more sense in a second. So what is going to be the color of these dots? Well, we'll make these ones red because this is our expected yield. So this is what we want to see most clearly. CX equals 1.5 just says like I want to make them, I want to make this window like 1.5 times bigger than normal. And then we want to define our y limb value, so how big our y-axis is going to go to, and we're going to define our y limb value based on our data. So we're going to say that our y limb value is going to be from 0 to the maximum value of the expected yield second column, which is the yield. Also, so this is saying the max value of the expected yield or the spread in yield, whichever one is greater, and whichever one is greater, I'm going to add 150 to that greatest value. And then the last thing we're going to do is we're going to say type O, so that means that it's going to plot as dots. And yeah, so that's it for that one. So remember, this was the expected yield. So this is the mean of the yield that we expect to get based off of the different HVEC values. Now we also want to see the spread in the yield. So we're going to plot that by using the points function. And so now we're saying spread and yield, the first column, which is the harvest rate, and do that by spread and yield second column, which is the yield. And then we're going to do the same, just housekeeping things for graphing, visualizations, the color of the spread. This will be blue. And again, we're gonna do type O. So it makes points. And then the last thing we want to do is we want to create the legend, so that way we know what these different things mean. And so we're gonna put the legend on the top right. And then the legend is going to say that our first one, our first series, the red series, is our expected yield, and the second series is our variability. And these are denoted by the red color and the blue color. Okay, let's run it. Let's see if we got it. So this could take a minute. Oh, here we go. So basically we want it to be, our optimal harvest rate is where our expected yield is highest and our variation is lowest. So that is clearly, I would say 0.25, because this is 0.05, 0.15, 0.25, 3.5, 4.5, 6.5, or 5.5, 6.5, 7.5, 8.5, 9.5, right? And so 25% is our optimal harvest rate. Now, what you guys will be doing for your homework is you will be looking at the bluefish tuna, and you're going to change the per capita growth rate to 0.286, and you're going to change the R spread to 0.2. So lower birth rate, but also lower spread. So should be around similar to our imaginary fish. But you're going to look at a much larger population. You're going to look at 100,000 versus 10,000. And this time you're going to get a very precise harvest rate that is optimal, because you're going to change your HVEC from 0.5 to 1 by 0.01. So this is what you're changing. And then once you've made those changes, screenshot an example of your graph, paste the screenshot. So for your homework, you want to paste the screenshot of the example graph you made in this code. You also want to paste a screenshot of your homework code, and then define what is the optimal harvest rate for the bluefin tuna based off your graph. Also, what is the optimal harvest rate for the theoretical made-up fish in the example problem? So we already found that out. That was 25%. And then, is the optimal harvest rate for the bluefin tuna lower or higher than the theoretical fish? Why? What parameters did we change that affected the harvest rate? So basically think back to these values, and if the harvest rate is higher or lower, why could that be based off of these parameters? Okay, it's been great showing you guys how you can use RStudio to visualize biological data sets. Feel free to reach out if you have any questions.